Okay, hello to all of you. I'm going to give you the instructions for the subcell lab, which we are going to do next week. So basically, first of all, we need to understand what the subcellular fractionation means. So let me go into the slideshow mode. So basically, this is a technique by which we can separate the different organelles. So as we know the structure of the cell has different organelles. The biggest organelle is nucleus, then we have mitochondria, then we have lysosomes, Golgi apparatus, and smooth ER and rough ER. So basically what we are trying to do in this lab is we are going to separate nucleus and mitochondria. So the technique which we are going to use in this lab is called differential centrifugation, and we are going to use different centrifugation speeds, and we are going to isolate first nucleus and then mitochondria, because these different organelles, they have different densities, and we can settle them down using different speeds, and this technique is called differential centrifugation. So the purpose of differential centrifugation is to separate subcellular components using a series of increasing various speeds. So basically, when we use the low speed, the more dense organelles will precipitate, and when we use the higher speeds, less dense organelles will precipitate. So I will be discussing the protocol with you, then you can understand. And then after we have done that, we need to confirm if we have actually isolated nucleus and mitochondria. So the aim of this experiment is to isolate nucleus first and then to isolate mitochondria. So you already know about what is nucleus, what is mitochondria. So basically, so this is the overall experimental design. So you will be working with cauliflower, you will be following the protocol, and you're going to basically make a cell suspension of the cauliflower cells in the buffers which has been given to you, and you have to make a very nice homogenate. It's very important to make a very nice homogenate, because otherwise you will have trouble in isolating these organelles. So you are going to make the homogenate, and then you're going to spin it down, and when you spin it down, you will be using initially the speed of 1000 G for 10 minutes. And remember that everything needs to be kept on cold. Even the centrifuge will be used at a cold temperature. So initial first spin will be, you will be taking the homogenate and you will be spinning it down at 1000 G for 10 minutes. And when you settle down, when you spin it down for 10 minutes, you will get a pellet. We will call it a P1. And in this pellet, we expect a lot of nucleus. And then you have this supernatant. This is called S1. So you will be taking this supernatant and you will be saving it also. I'm going to go over the detailed protocol also with you. You'll be taking this supernatant and you will be basically spinning it down again by using a speed of 10,000 G for 30 minutes. And then again you will get another pellet that we will call P2, and we expect a lot of mitochondria in this pellet, and then you will have S2. So basically you will be saving all these fractions. So we are not going to do all the different centrifugations. We are just going to limit our experiment to two spins. First one is at 1000 G for 10 minutes, and second one is at 10,000 G for 30 minutes. And in this way, you will get two supernatants and you will get two pellets. So P1, we expect to have a lot of nucleus, and P2, you will expect to have a lot of mitochondria. So we will be doing all our assays on all these fractions. So the first one is your filtrate, basically. The homogenate you make, we call it a filtrate. Then you have 5 fractions: filtrate, P1, P2, S1, and S2. And I'm going to discuss with you what are you going to do with all these fractions. So basically, the aim is to see a lot of nucleus in P1, and we are going to do a kind of a staining, which is, for this you will use a dye called Azure C, and that will stain your nucleus. And then we expect a lot of mitochondria in P2, and for that we will be doing one assay which is called SDH assay. It is an assay which depends on the presence of the enzyme called succinate dehydrogenase, and I will explain to you how you're going to do that, because this is an enzyme which is present in the inner membrane of mitochondria. If this enzyme is showing activity, it tells us that mitochondria is there. So you will be using light microscope for your staining for the nuclei, and you will be able to see these kind of nucleus. Okay, and I will be able to show you more pictures in the lab. So this is all nucleus. You can see how the nucleus will look like. And what are your expected results, like the homogenate or the filtrate? The first one, when we make the homogenate of the cells, we expect a lot of nuclei and many mitochondria in that because it has everything. When you spin it down in the first pellet, we expect many nuclei and some mitochondria because a lot of nuclei have settled down in P1. In S1, we expect few nucleus and many mitochondria. Then you will take the supernatant one and you are going to spin it down, and then you are going to get pellet number two. In pellet number two, we expect a lot of mitochondria and few nuclei. And in the S2, because everything has now settled down, the nucleus has been settled down in P1, mitochondria has been settled down in P2. In S2, we expect very few mitochondria and no nuclei. So these are our expected results. Yeah. So this is the essay basically you will be doing, which is for the presence of mitochondria. For the presence of nucleus, you're going to do the azure sea staining. It's very simple. You will take your samples on the drop of sample on the slide, and you are going to put the azure. You're basically going to make a wet mount, and you're going to add the dye, put the cover slip, watch under the microscope. And I showed you the pictures. The assay for mitochondria is called succinate dehydrogenase assay. This is an enzyme which is present in the inner membrane of mitochondria, and this enzyme is involved in Krebs cycle. We are going to talk about all these things in chapter number 10 in the classroom this week. In the Krebs cycle, there is one step where the succinate is converted into fumarate, and this is basically oxidation of succinate to fumarate. So oxidation, as you know, is a removal of electrons. So these electrons, when they are removed from succinate, where these electrons go? These electrons are taken by the electron carrier, and the electron carrier is FAD, and FAD is a coenzyme, so which means that FAD is part of this enzyme called succinate dehydrogenase. So when the succinate is converted into fumarate, this is oxidation. The electrons, they are carried by FAD, which is actually a coenzyme of succinate dehydrogenase, and this enzyme is present in the inner membrane of mitochondria. So this enzyme is also we will talk about all the four protein complexes which are present in the inner membrane of mitochondria. There are four protein complexes which are present in the inner membrane of mitochondria, and the second complex is actually the this succinate dehydrogenase enzyme only basically, and this is part of the four. There are four protein complexes, and these four protein complexes, they actually form the electron transport chain. So all the high energy electrons which are coming from the food, and they are being brought by the electron carriers. We have talked about all these things in the class. They are being brought here in the inner membrane of mitochondria, and eventually these all high energy electrons will be flowing through these protein carriers which form the electron transport chain, and eventually they will be delivered to oxygen, because oxygen is the final electron acceptor. So this is the way how these electrons, they flow through the electron transport chain, and finally they are delivered to oxygen. So what we are going to do in this experiment is, because we are trying to understand if in our fraction P2 specifically mitochondria is there or not. How to detect mitochondria? That will be detected by the presence of this enzyme called succinate dehydrogenase. If this enzyme is there, it is an indication that mitochondria is there. Now, how to detect that? So basically, as I just said, all the high energy electrons, they're eventually given to the oxygen, because oxygen is the final electron acceptor. So what we are going to do in this experiment is, we are going to block the path of these electrons to the electron transport chain, which eventually gives to oxygen. And how are we going to block it? We are going to use sodium azide. It is an electron transport chain blocker. So when the succinate is oxidized, the electrons are given to FAD, and FAD gets reduced to FADH2, and succinate gets converted into fumarate. And this reaction takes place with the help of the enzyme succinate dehydrogenase, because FAD is actually the coenzyme of succinate dehydrogenase. And then the electrons from FAD are given to electron transport chain and eventually given to oxygen. So this is normally happening. But what we are going to do in the experiment is, we are going to use sodium azide, and sodium azide is a electron transport chain blocker. and it will not let the electrons go to the electron transport chain and eventually to oxygen. So when the electrons are being blocked, then where these electrons are going to go, we are also going to use another indicator, which is called DCIP. And this DCIP is basically, so this DCIP is actually, when we use sodium azide, the path of electrons will be blocked. They will not go to electron transport chain. They will actually go to this DCIP, which is our redox indicator. So the electrons will be taken by DCIP, and DCIP is actually blue in color, and when it accepts the electrons, it gets reduced, and when it gets reduced, it gets light in color. It becomes light blue or colorless, and we can detect the change in color by using spectrophotometer, and the OD we will be using will be 600 nanometers. So this is the assay we are going to do, and this is a chart which is given in your protocol. I'm going to discuss that, how you are going to set up the experiment. So this is overall, I gave you some brief overview of the experiment, and now let me go to the protocol. So what you are going to do in this experiment, you are going to basically, here is some information given about some controls we are going to use. I will be talking those also to you, and this is a safety measure. Azide is toxic, so you need to wear the gloves. Okay, and yeah. Dispose all azide containing samples into the azide waste container and not down the sink drain. So more instructions will be coming in the lab. So you need to be careful when you're using azide, because it is toxic. Okay, so let's go over the protocol. So basically, as I said, everything has to be done on cold. Ice is very important. So collect the mortar and pestle and an ice bucket. So fill the bucket with ice and place the mortar and pestle on ice. You have to keep the mortar and pestle on ice. Use a razor blade to dice 20 grams of cauliflower. So cauliflowers are your plant cells. So we are working with the plant cells. Each group will need 20 grams of the tissue. Place the tissue in chilled mortar with 40 ml of ice cold mannitol grinding buffer. Grind the tissue vigorously with the chilled pestle for 4 minutes. Now here is a very important precaution, that this homogenate has to be made very, very nicely, because if this is not made nicely, then you will not be able to isolate nucleus and mitochondria. So be patient here, spend some time, make a very nice homogenate, keep grinding your cauliflower cells with this buffer. Okay, after it is done, filter the suspension through 4 layers of cheesecloth into a chilled 50 ml centrifuge tube. And this, so you will be basically filtering your suspension, because any cells, any part of your cauliflower which has not gone to homogenate, it should not be becoming part of your experiment, and we want to remove it, and it will be all filtered through the cheesecloth. So this liquid is known as filtrate, and this is our F fraction. Save 4 ml of filtrate into separate 15 ml falcon tube for later use. Place on ice. Remember that all the fractions, everything has to be done on ice in this experiment. So you will be saving around 4 ml of the your F fraction. Okay, and now what you're going to do, now you have this all homogenate and you have saved some. Now you are going to take this filtrate, you're going to centrifuge it. Centrifuge the remaining filtrate at 1000 G for 10 minutes at cold. Make sure the centrifuge tubes are balanced. So I will be there to help you with all these things. So the first spin will be done at 1000 G for 10 minutes. And remember that it has to be on cold. So for 10 minutes, you'll be spinning it down. So after 10 minutes, you will be collecting your tubes, and the whole class will be working together. All the groups will be working together. And after 10 minutes, you will get your tubes. You will see there is a pellet at the bottom, and then there's a supernatant. Now this pellet, we call it P1, and the supernatant is called S1. So basically, again, you have to take out this supernatant in a separate tube, and again, you will be saving 4 ml of S1 into 15 ml falcon tube. So these are our fractions which we are saving. We have already saved till now 4 ml of F fraction, 4 ml of S1 fraction. Now let's see what we are going to do with the residual S1 fraction and what we are going to do with the P1. Now the P1 is a pellet where we expect a lot of nucleus. So you have to resuspend P1 using a Pasteur pipette, and you have to resuspend into 8 ml of ice cold mannitol assay buffer. So to resuspend P1, use a pasture pipette, scrape the pellet from the wall of the centrifuge tube, and then with a pasture pipette, thoroughly resuspend this in 8 ml of ice cold mannitol assay buffer. So there are two kinds of buffers. One is mannitol grinding buffer, another is mannitol assay buffer. Okay, it is very important clumps be completely dispersed. So everything has to be on ice. Now what you have to do, now next what you have to do is with the leftover or the residual S1, you are going to spin it down at 12,000 G for 30 minutes at 4 degrees Celsius. So now the second spin is for a long time. It's very high speed and long time. Okay, now you have 30 minutes. In between 30 minutes, what are you going to do? You have F fraction, you have S1 and P2. With all these three fractions, you are going to make a wet mount and you're going to put the azure sea dye on these and just cover them with cover slip and watch under the microscope. And you will be able to see the nucleus. So that's what you have to make your observations. I already showed you how the nucleus will look like, and I will show you more pictures in the classroom, in the lab, sorry. So this 30 minutes you will be using for that. And after your spin is done, what are you going to do? Now you will again see you have a pellet at the bottom. This pellet is P2. And in this pellet, we expect a lot of mitochondria, and it should give you appearance like little brownish because the mitochondria has these special proteins called cytochromes. Because of that, they should have some dark color. And then you will have a supernatant. That supernatant we call S2. And again, save 4 ml of this supernatant. And actually, we are not going to do anything beyond this. Now we have 5 fractions with us. We have F, we have P1, we have S1, we have P2, and we have S2. Now what we have to do, we have to basically do the SDH assay. So for that, what you have to do, for that this table is given. And remember that the wavelength which you have to measure, it has to be at 600 nanometers. So your filter should be on the right side, and you have to warm up the spectronic. So this is the table according to which you are going to make your cuvettes. So remember that for every fraction, so you have 5 fractions. For every fraction, you have to make the blank. Okay. Now, what is a blank and why we are making it? Okay. So let's go and discuss this table. Now you have 5 fractions. Okay, what is a blank? Blank is for every fraction you have to make a different blank. Blank is something with which you are going to calibrate your spectronic. It's a blank for every fraction. Now blank will have everything except DCIP. Now what is DCIP? It's the artificial electron carrier, electron acceptor, sorry. It's an artificial electron acceptor. So from where does it accepting the electrons? Because we have used sodium azide. Sodium azide is going to block the flow of electrons to the electron transport chain. Instead of electrons going to the electron transport chain and eventually going to oxygen, the electrons will be accepted by DCIP, which is the artificial electron acceptor. Now, DCIP is blue in color. When it accepts electrons, it gets reduced. Its color becomes light blue. So which means that if the DCIP is getting reduced, its color will become from blue to light, and its color should become blue to light. And if that is happening, it means that the SDH is working. SDH enzyme is there. And if SDH enzyme is there, succinate dehydrogenase is there, it means mitochondria is there. So now let us see the setup of the experiment. So for filtrate, you will be adding 1.04 ml of the mannitol assay buffer. Then azide is 0.2 ml, DCIP is 0.2 ml, succinate is 0.2 ml, and your fractionation, this is the first one is for filtrate, 0.36 ml. Okay, and the blank will have everything except DCIP. We do not want DCIP here because it's blank. You have to calibrate the spectronic with this. And because we are not adding DCIP, we are adding little additional buffer here. You can see more buffer is here. Okay. Now with the blank, you are basically going to calibrate your spectronic, and with your filtrate fraction, you are going to add all these things, and you are going to measure the OD at 0 minute as well as after 3 minutes. It's a very important because we are giving the time for the DCIP to get reduced, and the reaction time is 3 minutes. You have to take the OD at 0 minute, and then you have to take the OD after 3 minutes. And then the same thing you have to do with the S1 fraction, P1 fraction, S2 fraction, and P2 fraction. So you have to add all these volumes. Remember that for every fraction there's a blank. Blank we are using for calibrating our spectronic. The blank does not get DCIP. So with the blank you calibrate it, and with the real fraction, with your real whether it's a filtrate S1 P2, you will be taking the OD at 600 nanometers at 0 minute, and then after 3 minutes. So basically we are trying to see if the OD is reducing or not. If the OD is reducing, it means that DCIP is getting reduced. If it is getting reduced, which means the enzyme is there. If the enzyme is there, mitochondria is there. And also in this experiment, we are going to put some controls. Now we are going to use a control called malonate. And when you set up all these controls, remember that you have to use your, like we are expecting a lot of mitochondria in P2. So P2 is our positive control, and you will be having a blank for that, and you will be having this fraction. This is our positive control, and then we are using some negative controls. So this you have to set up again. So this is basically the same thing. This for these controls you are using these again. It is good because you will have your results in duplicates. Now malonate control is, malonate is basically a competitive inhibitor for the enzyme succinate dehydrogenase, basically, which means that it is a malonate and succinate, they basically compete for the active site of the enzyme succinate dehydrogenase. So when you add malonate, malonate is a competitive inhibitor for enzyme SDH. So when you add malonate, then malonate will go and bind to the active site. Succinate will not be able to bind. When succinate is not able to bind, then you will not see the results, because the reaction which is catalyzed by this enzyme is the oxidation of succinate to fumarate. When succinate is not there, this reaction will not take place. So malonate is your negative control. You should see reduced reduction here. And then we will be also using azide as a control. So basically, what is the azide control? Azide control is that control in which we are not adding azide. So when we are not adding azide, what's going to happen? When there is no azide, then there is no blocking of the electrons to the electron transport chain, which means the electrons will not be passed to the, when the azide is not there, the flow of electrons will not be blocked, and the electrons will actually go to electron transport chain, and eventually they will be going to oxygen. So which means that when we use azide, when we do not use azide, the electrons are going to go to electron transport chain, and they're not going to go to the DCIP. So in this case also, you should see reduction of DCIP, because it is not going to get the electrons, because the path of electrons is changed now, because no azide, no blocking, electrons will go to their normal course. Then third control is succinate control. Now in the succinate control, we are going to skip adding succinate. So there will be no succinate. So if there is no succinate, then no reaction, right? No succinate, no fumarate. No, if there's no succinate, no oxidation, no electrons. Then eventually the reduction of DCIP will not take place. These are our controls. These are all our negative controls basically. So all the controls, why keep, so that we can see if everything is going. So this is OD. You find out the rate of DCIP reduction and how to do that. First of all, for every fraction, you need to know what is the difference of your ODs. So for every fraction, you are going to take the OD of 3 minutes, and you're going to subtract the OD of 0 minute from that. That gives you the change in the OD. Now, concentration of DCIP reduced can be calculated by taking the change in the OD, and you will be dividing by extinction coefficient for DCIP. The extinction coefficient for DCIP is 21 millimolar per centimeter. So this is the value. So you will be taking the change in OD, and you're going to divide by the extinction coefficient of DCIP, and that gives you the concentration of DCIP reduced. And this value will be in millimolars, and then after that you will be converting everything to the micromolars. So this is the concentration of DCIP reduced. After that you can calculate the amount of DCIP reduced, and that can be calculated by taking the concentration of DCIP reduced and you can multiply it by the volume of reaction. Now the volume of reaction is 2 ml. So that gives you the amount of DCIP reduced in micromoles. And then eventually you can calculate velocity or rate of DCIP reduction, and that can be calculated by the amount of DCIP reduced divided by the reaction time, which is 3 minutes. Okay, so first you will calculate the concentration of DCIP reduced, which can be calculated by change in OD divided by the extinction coefficient of DCIP. That gives you the concentration in millimolars. And after that, you can calculate amount of DCIP reduced, and that can be calculated by the concentration of DCIP reduced, which you have calculated here, and multiplied by the volume, which is 2 ml. This gives you, you have to calculate in micromoles. This is the amount of DCIP reduced. And then the rate of DCIP reduction can be calculated by taking the amount of DCIP reduced and divided by the reaction time. And the reaction time is 3 minutes. So the velocity units will be micromoles per minute. So this is what you have to do, and these are 5 questions you have to answer, and we can discuss in the class if we have time. So that's all about the instructions, and I will be working with you guys in the lab.